DNA sequencing. Determine precise order of nucleotides along genome. By denature DNA for fragmentation and library preparation. For sequencing. To study genes' structure and functions and genetic variation. Whole genome sequencing is a type of DNA sequencing. Determine complete DNA sequences in genome at single time, as chromosomes and mitochondria and chloroplast in plants. For comprehensive analysis of entire genome and study variants in coding regions, as mentioned in next slide. While target or gene sequencing is another type of DNA sequencing. Determine sequence of particular genes from whole genome sequences by using specific probes to capture interested genes in process of hybridization. For sequencing. To study genetic analysis in details, as mentioned in next slide. Whereas shotgun sequencing is a type of DNA sequencing. Determine sequence of large piece of DNA, as mentioned in the table inside slide, and figure in next slide. On the other hand, clone-by-clone sequencing is a type of DNA sequencing. Determine accurate sequence of various chromosomes at single time, using vectors, to generate multiple copies of each DNA fragment, as mentioned in next slide. Otherwise, pyrosequencing is another type of DNA sequencing, depending upon release of PPi and ATP, as mentioned inside slide. So, DNA sequencing initiate by DNA extraction and DNA fragmentation into smaller pieces by restriction enzymes, and adaptor ligation to each ends of DNA fragments. For library preparation and library enrichment by PCR, to generate quantities of DNA fragments with adaptors. For sequencing, using any of sequence platforms to align sequences to reference genomes, as mentioned in next slide.